I'd like to welcome you all to the Spring 2023 edition of the Broad MIT Seminar Series in Chemical Biology. To introduce the series, this started just before the pandemic when chemical biologists at MIT, Laura Kiesling, Matt Shoulders, and myself crossed over Main Street and started talking to chemical biologists at the Broad Institute, Amit Choudhary and Bridget Wagner in particular. And we brainstormed and designed a seminar series that would be attractive to the scientists at both institutions. And of course, this leverages Kendall Square being the epicenter for chemical biology worldwide. Part of that epicenter is MIT, part of the epicenter is the Broad Institute, which of course includes faculty from both MIT and Harvard, but another key part of that epicenter are the fantastic pharmaceutical companies and biotechnology companies that also are in our environment. And they have been wonderful to interact with, and the companies that you see here on the slide have contributed to this series by sponsoring these lectures. So we're very, very grateful to these companies, Biogen, Merck, Novartis, Pfizer, and Roivant in particular. So we have had a wonderful lineup in the fall, and you can see 3 of the speakers from the fall. In the spring, we have 7 speakers starting today, so I hope you keep coming back to hear these wonderful talks from these, some of the leading chemical biologists worldwide. And I'm going to now pass the microphone to my colleague, Xiao Wang, who personally bridges across Main Street. She is a faculty member. She's my colleague at MIT, but her research laboratories are here at the Broad Institute.
Good afternoon, everyone. I'm Xiao Wang, an assistant professor joined between MIT Chemistry and Broad Institute. Today is my great honor to introduce our speaker, the first speaker in 2023. So Dr. Chuan He is a John T. Wilson Service Professor at the University of Chicago Chemistry. And in case you don't know, he's also my PhD advisor. So I'm super excited about his visit here. So Chuan is best known for his work in the study of DNA and RNA chemical modifications, especially their role in cancer epigenetics. Recently, his research is also extended into agriculture applications, which is super exciting. You will hear the story during his talk. And I'm supposed to tell a bit more about Chuan besides what you can find from Wikipedia. I think besides a great pioneer, a scientist, Chuan is also a great mentor. I still remember when I was a graduate student in his lab, sometimes I got frustrated with all the negative results. And whenever I walk into Chuan's office after Chuan seeing these pretty unexpected negative results, he always say, that this is even better. So I feel so encouraged through all this interaction with him, and from which I learned science is not about success or achievement. It's really about stay open-minded and stay positive about all these unexpected, exciting discoveries. So here, welcome, Chuan. Yeah.
I thought it's not a place to share stories. One of the accomplishments I made was when Xiao joined the graduate school, she was interested in organic material. And I think one of the accomplishments was converting her to study biology these days. All right. Well, I was thinking about what should I present today, and unfortunately, 2 weeks ago, Laura and I were in Paris. So she heard all my talk. I decided to give you a slightly different version. So the first part, I'm going to actually present some of the newest discoveries in RNA modifications in transcription chromatin regulation. It's going to be quite biological. But then the second part, I'm going to actually move to biochemistry and chemistry. So this is really, I hope, a lecture for everybody. Sort of reflect back to my roots. I was a graduate student here working on bioinorganic chemistry. The whole thing started with metalloenzymes. I was actually working on this these ion-dependent alpha-ketoglutarate demethylases. These are the ones that oxidizes the methyl groups on the nitrogen, and you can see, and that leads to production of a hemiaminal, and as we know, this is not stable in water, fall apart to give you formaldehyde and repair the base. So this was a way to perform DNA damage repair in E. coli. So this was my first job proposal. We got it done, and we start to move along, and then we realized in human, there are 9 human homologs. Right? So we worked on this. We got interesting results, papers published. I got my tenure. At one point, we wondered, why there are 9 of them, right? Only 1 or 2 of them perform DNA damage repair, just like in E. coli case. So back in 2010, we sort of proposed this idea that lots of these human homologs not only work on DNA, or they probably don't work on DNA, majority of them should work on RNA, because all you need is as a methyl group on the nitrogen, these enzymes will oxidize that methyl and leads to demethylation. Right? So this was a hypothesis back in 2010. It turns out that ended up to be the case. One of my former postdoc, Guifang Jia, showed in this 2011 paper that if you take FTO, one of these human homologs, mixed with iron alpha-ketoglutarate in the presence of oxygen, biochemically, you clearly see a demethylation of M6A. So this base, this methylated base is getting demethylated. Right? That was the first clue in the lab. And what is this M6A? It's N6-methyladenosine. It turned out I have not heard about M6A, unfortunately, before 2010, right? I heard about this in 2010. We started to test some of these. This is the most abundant internal modifications for mammalian messenger RNA. It's conserved in many eukaryotic messenger RNA. And in average, every messenger RNA in human cells, mouse cells contains 3. And it's essential for mammals, conserved in lots of eukaryotes. It was discovered back in 70s, but because of lack of technology, lack of molecular cloning, we didn't really know much about this, except we know this is very important. This is the substrate for that demethylation. Turns out, since its discovery in 1970s, for 40 years, people know this is the most abundant modifications on the messenger RNA, and there's one enzyme, METTO3, installed this. That was it. So we came in, made this unexpected discovery of a demethylase FTO, and a couple years later, we showed the second demethylase, ALKBH5. Both are critical with major phenotypes, FTO being essential in humans, ALKBH5 essential for spermatogenesis, suggesting there's a program built in for regulation through this RNA methylation. The methyltransferase is a big complex, over 1,000 kD. We and others characterized later on. I'll come back to this, very interesting. Now, the reason the field really took off, back in 2011, that was the only paper. If you check Google Scholars, probably several thousand papers on M6A last year. There's 2 reasons. One, these modifications are key for every stem cell differentiation and tissue development. For every cell differentiation, it's critical. The second reason, really because of the discovery, characterization of a series of proteins that preferentially recognize methylated RNA, regulates the decay, stability, translation, localization of this methylated RNA. So the YTH family containing proteins, these are proteins containing a domain, evolved to preferentially bind methylated adenosine. So there are 5 of those and other proteins. Right? These were actually the subject I tend to present in my regular talk. But as you probably noticed, these were done by my host here, who just introduced me. So I decided not to embarrass her. So I'm not going to talk about this work. I'm going to actually talk about something else. So it's really Xiao's work laid down this foundation for YTG-DF2, DF1. I think each of these papers probably cited over 2,000 times or 3,000 times now. I'm going to come back to this demethylase and actually share with you something about this demethylase. Started from 2011, we're still working on it. It's totally unexpected. Every time we learn something different, right? So this demethylase, as I just introduced to you, biochemically, you see this complete demethylation when you mix with the methylated RNA. And in cancer cells, this is the HeLa cells, you can see you can purify poly-A RNA. And if you knock down FTO, you see an increase of M6A, right? So it's about 15% increase. And that indicates inside of the cells also a messenger RNA demethylase. There has been lots of confusion about the exact physiological substrates. There are multiple layers of confusion. First, is M6A the real substrates, right? They were hypotheses or paper published 5 years ago suggesting works on the cap, M6AM. So it turns out after the cap, if the base is A, and that one can be methylated at 2' position as well as the N6 position. And this one was shown to be a major substrate of FTO, and that's the hypothesis. This is the one drive all the biological function, right? Turns out this is a very good substrate. It's about 10% of M6A in terms of the abundance, but we and others showed that in the last 5 years, there's enough results showing that this is interesting. We do not know the biological significance yet. Probably interesting in immune or neuronal context, but in majority of the cancer cases, other cases, the internal M6A is the key substrates that drive the biological consequences. Another major confusion or another mystery in my lab, we care most, is what's exactly going on with FTO under physiological conditions. In cancer, we know it works on messenger RNA extensively. However, in primary tissues, in mouse and human, it's not the case. In neurons and the adipocytes, FTO works on messenger RNA. You can clearly see that. But in other primary cells, tissues, if you isolate knockout tissue versus wild type, you do not see that much messenger RNA demethylation. There are other work suggesting messenger RNA may not be the primary substrate, right? However, if you knock out FTO, the mouse died late embryo stage. Those who were born have developmental defects. And this is actually lethal in human. So we don't see the mutant in human, right? There's one mutation, real mutation, leads to about loss of 90% of demethylation activity. The infant can be born, they have severe cardiac and neuronal defects, and they typically don't pass year 1. So we saw a few cases in New York City. We have isolated, we generated iPS cells trying to study. So people don't really know the physiological substrates of this one. Going back to M6AM, if you look at the methyltransferase knockout of M6AM, they develop perfectly fine. So the writer for M6AM doesn't have developmental phenotype. The eraser works on part of this, should not have any physiological, should not have, this should not be the substrate account for this lethal phenotype. So what's really going on? Well, that will lead to my first part of presentation, which goes beyond this post-transcriptional regulation. In the last 8 or 9 years, 99.9% of the work on the RNA methylation field been focusing on the post-transcriptional regulation. But today I'm actually going to share with you something else has been going on. This major big methyltransferase complex is not only install methylation to pre-messenger RNA, but it turns out it installs the methylation also to these non-coding RNAs associated to the chromatin. These are promoter-associated RNAs, enhancer RNAs, and the repeat RNAs. We had this hypothesis back in 2018 that the methyltransferase might be doing something beyond messenger RNA. So the first experiment we decided to do with my former postdoc, Jun Liu, when she joined the lab, we just asked the methyltransferase knockout the mouse embryonic stem cell from Howard Chang's lab. I did a quick experiments, look at the global transcription difference and global chromatin difference. So this is the EO labeling to look at the total output of nascent RNA. As you can see, if you delete methyltransferase, the entire transcriptome is upregulated, right? There's a lot more nascent RNAs produced. You can rescue this with a Y-type methyltransferase, but not an inactive mutant. You can also perform tunnel assay. This is essentially DNA's digestion, and you generate all the ends, and then you label it with fluorescence group. So the more bright will indicate more opening of the chromatin. As you can clearly see, if you knock out the methyltransferase, the chromatin become more open. Again, you can rescue with Y-type, but not an inactive mutant. What this indicates, the methyltransferase somehow play a critical role to the chromatin openness. We went on isolated chromatin fraction, performed M6A sequencing, so you can immunoprecipitate M6A modified RNA. You have a calibration probe. You know exactly where the methylation occurs, the relative level. And as you can see that, indeed, if we deplete the methyltransferase, we see a reduction of methylation on the chromatin RNA. And if you exclude the messenger RNA, lots of these enhancer RNAs, promoter RNAs, repeat RNAs, you see reduction of the methylation. Now you ask, what's going on with this RNA on the chromatin? Only on the chromatin, not the whole cell, right? Now the absence of methylation leads to more of these RNAs on the chromatin. They're more stabilized. The presence of methylation leads to depletion of this RNA on the chromatin. We then quickly switch to another reader protein. This is YTH-DC1. This is also embryonically lethal, very embryonically lethal. There are 5 YTH family proteins. This is the one we didn't study in early days. This is very important. It sits in the nucleus. And if we delete it, we made a conditional knockout in mouse embryonic stem cell. You can see more nascent RNA production. We can rescue this with a Y-type, but not a mutant that won't recognize the methylation. And if we look at the tunnel assay, the chromatin openness, again, if we deplete this reader protein, we see more open chromatin. We can rescue with Y-type, but not a mutant, which will not recognize M6A. So a very, very simple scheme. We have this some sort of RNA stick to the chromatin that can be methylated. This is not coding. The YTH-DC1 is a reader protein, recognize the methylation. Somehow that leads to decay of this RNA. So very quickly, again, I just going to go through this genomic work. We take YTH-DC1 knockout embryonic stem cell Y-type. We look at the chromatin fraction, sequence RNA, ask what are the RNAs that are methylated, what are the RNAs that are manipulated by this protein. We realized the enhancer RNA, promoter RNA, we did not see difference on the methylation. However, for this repeat RNA, we see major increase of M6A. So the YTH-DC1, the reader protein, the methylation occurs to all these different RNAs, but the repeats, the ritual transposon RNAs, are the ones that are decayed through YTH-DC1, because you knock out YTH-DC1, you see increased M6A. And then you ask, what are these RNAs? Line 1, I'm going to come back to this. ERV, all these ritual transposon RNAs, they are the substrates of YTH-DC1. And you can actually do experiment to confirm this. This is the Y-type decay of that RNA. If you inhibit transcription, you look at the chromatin fraction, the RNA goes away. But if you deplete the methylation, or if you just get rid of YTH-DC1, you see delayed degradation or decay of this. Right? So what's the consequence of this? Why this leads to global chromatin openness? It turns out there's about 6,000 genes. 6,000 genes in mouse embryonic stem cell Y-type have upstream RNA methylation. These methyl groups play critical roles to the transcription of these 6,000 genes. If you get rid of methylation, 99% of the case, these genes are upregulated, chromatin become more open. Right? So it's really a global and a local change. Mechanistically, I haven't told you why this is the case. It's turned out many of these RNAs are critical to assemble transcriptional factors, recruit histone modifiers to activate transcription and add active histone marks. So here you can see that if you get rid of methylation, look at the global level of K4 trimethyl, K27 acetylation. These are active histone marks, you can see global increase. The entire histone level remains the same, but then the active marks accumulating, right? So both locally and globally, if you look at the 6,000 genes, look at the transcription starting sites, you can see this is the Y type, the gray, and then the yellow ones are the methyltransferase depletion. If you get rid of methylation out of these 6,000 genes, if you look at transcription starting site, there's just more active histone marks. There's just more open chromatin. I already told you that the presence of in the last, I would say, 5 to 8 or 10 years, there's lots of research ongoing to show the importance of these RNAs on the chromatin. They're critical in terms of assembling transcription active complexes. Some of the other speaker, Rick Young work, YY1, transcriptional factor, well known to be assembled on these RNAs. And CBPs or these histone modifiers, the presence of RNA recruits this to assemble. You probably heard of phase separation, condensates. RNA play a critical role, not only in the cytosol, but also in the nuclease to assemble these hubs. They also repair the histone repressors, such as PRC2. So this is known, but there's a lot of biology behind this. We still don't know that much about the roles of this RNA. Anyway, the bottom line is we have these non-coding RNAs play critical roles. I think there's going to be a lot more work. There's like every week there will be paper coming out to suggest important roles of these RNAs. They play critical roles to regulate the local and the global chromatin state. They are regulated through modification. Okay, you can methylate them, and then that methyl group can be recognized by a reader protein. Then there's a two outcome. The reader protein can recruit proteins to decay the RNA, or they can recruit proteins to add suppressive marks nearby. The effect of the methylation is to lead to more closed chromatin. To confirm this, we constructed a demethylase CRISPR system, and we look at one super enhancer. Here's a methylation site, and if you knock out the methyltransferase, so here's the methylation. You knock out the methyltransferase, the methyl group, the methylation level reduces, and the open chromatin in the active histone markers increase, indicating a more open chromatin state. If you deliver this demethylase to that particular methyl group in the super enhancer in the Y-type mouse embryonic stem cell, you see a reduction of M6A, increased half-life time, increased the transcription rate, and increased the histone marks, indicating open chromatin. So you can, we did this to 10 different sites. You can really connect the methylation to the outcome of transcription. So that's sort of serve as an introduction. What we essentially showed in this 2020 paper for the first time, the methylation not only impacted the messenger RNA fate, they're really important to the chromatin regulation. And we were not the only one thinking about this. 3 labs published back-to-back papers a year later, pretty much confirmed our work, but pushed this much further, indicating that not only decay is regulated, but these methyl groups can be recognized by readers to recruit complexes that modifies the histones and change the chromatin state. Now, that actually set up something we started to think about related to our demethylase, right? So for a long time, right, people in Cambridge and elsewhere, or epigenetics, we know DNA modifications, histone modifications are critical to chromatin regulation. Unfortunately, now we have to add RNA, right? Not only I showed you the writer, the methyltransferase, but also reader, the YTGDCY, right? The question is, are these processes reversible? Actually, could some of the demethylase, the real physiological substrates, are this car-RNA instead of the messenger RNA, in particular, FTO, right? I introduced to you that if you knock out FTO, you have this quite severe phenotype. Most mice die at late embryo stage. We know if we knock out the methyltransferase, most embryo die at early embryo stage is quite essential. So could FTO work on a subset of METTO3 substrates, right? METTL3 installs messenger RNA methylation, but it also installs this chromatin RNA methylation. So if FTO does not work on messenger RNA, could FTO work on this chromatin RNA? So that was our hypothesis going into this work. So a previous graduate student stayed as a postdoc, decided to take this challenge and look at the FTO knockout mouse embryonic stem cell. When you isolate the messenger RNA, you see there's a slight increase, not much. But if you isolate the chromatin fraction, you can see a notable increase of M6A, indicating that at least in mouse embryonic stem cell, FTO seems to work on this chromatin RNA. What are these chromatin RNAs? Well, you can perform a sequencing of the chromatin fraction in the absence and presence of FTO, and you analyze it, you realize the majority of the methyl groups, well, methyl groups occurs through enhancer RNAs, promoter RNAs, repeat RNAs, but when you delete FTO, the major change is on the retrotransposon RNA, in particular line 1. You see depletion of methyl, you see accumulation of methyl groups because you got rid of the erasers, and these line 1s become less stable, right? The methyl group leads to less line 1. It's a demethylase. You get rid of demethylase, more methyl groups on the RNA, and they're being destabilized. What is line 1? For people in the genomic field, it knows very well. For chemical biologists, probably not, right? Our genome encodes about 1.5% of our genome encodes coding protein, right? But majority of our genome are occupied by retrotransposon elements, right? Line element is one retrotransposon has jumped around in our genome 1.5 million times. Line 1 is about 400,000 copies, one, right? It's still active constantly, well, not constantly, in some of our cells, it's still activated, transcribed to become a protein, and then regenerate, reinsert back into the genome, right? It's active retrotransposon. This is lines, about 18% of our genome, right? Not all of this is transcriptionally activated. Part of those transcriptionally activated, those activated undergo demethylation, methylation and demethylation. The methylation is most likely a mechanism evolved early on to get rid of, to suppress these retrotransposons. Through evolution, they've been adapted for gene expression regulation. Again, I'm going to just go through some of this data quickly. We isolated the line elements from Y-type and FTO knockout. We clearly see accumulation of M6A and the reduction of the line 1 level. Now, not all line RNAs are substrates. The young active lines, I'm going to come back to this, are the substrates of FTO. And we did a bunch of experiments to show this is activity dependent. We can use a FTO inhibitor. We observe exactly the same. And when we sequence RNA-seq, if we knock out FTO versus we use ASO to block line 1, we see very similar expression profile indicating when we get rid of FTO, the effect comes through the line in general. What's the physiological relevance? There's probably another experimental results most people don't know. Line is so important. If you block a line, mammalian system were stuck at a 2-cell. If you block a line at early embryo in mouse systems, embryo stuck at 2-cell. It cannot propagate to 4-cell, 8-cell. It's that important, all right? In mouse embryonic stem cell, the line level goes up 6-fold upon differentiation. The FTO level goes up 3-fold upon differentiation. So these are very important for differentiation. Now, if you knock out FTO, the line level can only go up 3-fold upon mouse embryonic stem cell differentiation. That caused increased differentiation, reduced proliferation, a defect, right? And we can rescue this defect with FTO fused to DCAS13 system targeting line 1. We can recover about 80% of the 70% of the defect, indicating this is really one of the main substrates. Mechanistically, again, this is with 27 supporting figures. I'm just going to summarize, give you sort of a simple mechanism. What's really going on? There are lots of lines activated inside of the cells, but there are about 1,400 of genes in mouse embryonic stem cell contain line 1 in introns or at the antisense, so in the genes. these are the ones affected the most compared to other genes that contains line 1 outside of the gene, right? So it turns out it's really these guys. Our genes contain line 1 in the introns or antisense. They can be methylated. The methylated RNA will recruit the degradation systems or histone modifiers to suppress transcription. And if you get rid of FTO, right, the FTO antagonizes, remove the methyl group to activate gene expression. If you get rid of FTO, you accumulate methylation. That leads to close the chromatin and reduce the transcription. You compare this 1,000 to the rest, you clearly see the difference, and you can confirm this with histone sequencing, histone mark sequencing, right? So that's what's going on. This is also true in mouse. If you take a FTO knockout mice, at very early embryo stage, you realize the same line elements that start to accumulate M6A, and these are the genes contain line in their intron or antisense. When you knock out FTO, these lines accumulate M6A, and they actually lead to reduce the level of these genes are critical for early development. We can actually inject the FTO Cas13 system to the early embryo and rescue these gene expressions during mouse development. So, all right, we're almost done with all the genomics. So from now on, will be easy, right? I try to just get through those, give you a sense what's really going on. All these experiments establish two things. Number one, methylation is critical to chromatin regulation, transcription regulation. Number two, FTO works on messenger RNA in disease samples, in neurons, in dipsites, but during early development, FTO is critical to regulate the chromatin state and the gene expression, okay? What about human and mouse tissue? Right? If you look at globally, the messenger RNA methylation versus FTO level in human and mouse tissue, you do not see a correlation. However, if you just look at the line 1, you take line 1 out, and you go through all the tissues in mouse and human, you look for high FTO tissue. When the FTO level is high, the line M6A level is low, and then the line level is high. You see a clear correlation in mouse, not so much in human, but still very good trend. This indicates line is the substrate in most tissues for FTO. Right? You don't see this for another demethylase, which works on messenger RNA, and we actually know that FTO accumulates mostly in brain and heart. We made a cerebellum knockout FTO. We can validate that, again, in cerebellum, if you get rid of FTO, you see accumulation of M6A, reduce the line 1, and these play quite interesting roles in cerebellum functions our collaborators been working on. So the bottom line, in the last 2 years, there's a new sort of paradigm of chromatin regulation or RNA methylation emerging, that this methylation play critical roles on chromatin regulation, right? You have writers to install this, affects thousands of genes. You have different readers recognize different types of methylation, regulate different sets of gene expression. We also have these erasers that comes in to reverse the process. We started to play with this. For those interested in embryogenesis, stem cell biology, these are really interesting. We have all kinds of cool phenotypes. We can grow mouse different ways we want by manipulating this, shockingly. But my former postdoc had a much better idea. And we will play with this. We're going to play with pig and cow. And my former postdoc said, well, let's play with plants, right? And I spent 10 years in China. I stopped because of our previous president, the current one. So, but I spent 10 years in China. So one thing I did was plant biology. I was lots of plant biology expertise in China. We decided to get into plant biology. So with the same postdoc, Guifang Jia, we've been working at plant biology. We discovered the first plant demethylases. And she had this crazy idea of let's insert human FTO into plants. We did that with Arabidopsis. We didn't see much. And one day I was at home. This other one, Professor Song, was the president of the local university. He turned out to be a plant biologist working on rice. And I said, well, let's just try this idea again in rice, and I completely forgot about this. A year later, I received this. So when we insert FTO into rice, we really drive the plant development. The root system expands. And we see about 3-fold increase of yield and biomass in greenhouse. Right? If we move to the real field in 2 different climate conditions, we actually see a 50% increase of biomass and yield. And these plants are more drought resistant as well. Right? So we kind of wonder what's going on. This work took 7 years. We take RNAs out of the rice and we perform the sequencing. For 2 years, we couldn't understand what's going on, right? Because there was no difference after you analyze the data. Until one day I was in Beijing, and suddenly something hit me. I said, well, why don't you isolate 1 gram of roots, measure the total amount of messenger RNA? Student came back, said, oh yeah, Professor He, you're right. When you insert FTO, the entire messenger RNA level increased by 2-fold, right? So the entire transcriptome is upregulated. When you insert the FTO, the entire chromatin opens up. Remember, we knock out FTO, that leads to a closed chromatin. If you insert the FTO, it's like you dropped a nuke. The plant somehow has an intrinsic pathway to use chromatin RNA methylation to control its openness and transcription. If you overexpress FTO, you dramatically open the chromatin, all the transcription elevated, right? The way we typically do RNA-seq is we normalize them to the means, right? If you normalize to the means, you won't be able to do it. All you need is add the calibration, and you see the entire transcription, most genes upregulated. And that somehow leads to a lot more proliferation at the root system. So there are meristem cells at the tip, and there's lots of proliferation compared to the Y-type. We believe that was the reason to drive this root development. Also, at the top, we believe that leads to more branch, and that's more biomass. Right? So Xiao and I actually eat rice a lot more, right? But for some of you, here's your potato. Right? I actually deleted another slide because we actually have trees, grass, and other things working. And in my lab, we actually grow the field of plants. And the other day, I was taking picture. Myself didn't recognize what type of plant. That was embarrassing. But they grow bigger. Yeah, so we're having a lot of fun. We'll see how this will lead us. I learned the yield is one part, but really the drought resistance is a major part because of climate change. All right, so that's the part Laura heard 2 weeks ago. Now we're going to actually move on to the chemistry part. I thought I'm going to introduce you some of the methods we've been developing to map RNA modifications. Now, the current way we sequence RNA modification mostly use antibody, right? So you fragment the RNA into different pieces. The piece that contains the methylation will be enriched with an antibody, and we know in that 100 nucleotide there's a methyl group, right? That's it. There's no resolution. Won't tell you the stoichiometry, but it's been very useful. Everybody uses, right? This is what you get, right? And obviously, as anybody in biology or chemistry, in particular biochemists would like to know where it occurs, exact position of methylation, and the stoichiometry. There's a gold standard for that. When people study DNA methylation, there's a bisulfite sequencing. So what does bisulfite sequencing work on? It takes the bisulfite reaction, convert all the Cs to use a deamination. The methylated C resists the stays as a C. So you know this is a C when you perform sequencing before conversion. After conversion, only those stay as a C will be methyl C, right? So for the 20% of the methyl C, after the sequencing, you'll see 20% the C, 80% the T, all right? So that's how you know exactly where it occurs, modification fraction. So we wondered, could we do the same with A? Can we have a deaminase? I had this idea 80 years ago, never really worked until one day we hired one of the postdocs from Broad Institute who brought the deaminase from here. Adenosine deaminase further evolved. Weixin Tan has the deaminase people use for CRISPR, the base editor. similar group of deaminase are very potent. It deaminates all the A to inosine and doesn't touch the methyl A. So that's the way we can sequence, right? And my former student postdocs collaborated to develop a chemical method called GLORY, can do similar things. But these are sort of a negative readout. What about the positive readout, right? You have to do subtraction to figure out the M6A. Here's a positive readout. This is actually really some nice biochemistry. It was not my idea. One of my postdocs insisted she wanted to do this. I discouraged her 3 times. She still went ahead and did it, right? And it worked. So there's this ribosomal RNA dimethyltransferase, MJDIM. This one methylates A twice to generate dimethyl A. So my postdoc had this idea that for this, the enzyme obviously prefers the methylated one. Otherwise, it won't have this, right? So she proposed that if that's the case, we could use this enzyme and a SAM cofactor with a Leo group, right? We developed this previously. And maybe this one will prefer the methyl group, install a Leo group, and we know that if we treat this with iodine, we'll cyclize, and that will block Watson-Crick base pair. When you bring in HIV-RT, will read as a mutation, right? And when I look at this proposal, I said there's too many ifs, right? There's 2, 3 hypotheses. You have to be extremely lucky. First of all, this substrate has to work. Second, the enzyme would have to prefer that one. Third, you have to have enough mutation because methylated A is only less than 1% of A. This needs to be at least 50 to 100-fold selective. And to my surprise, she was extremely lucky. It worked. The Leo-SAM works quite efficiently. And as we would have predicted, the enzyme prefers the methyl group by about 10-folds. But that's not enough, right? That's not enough. She was extremely lucky. Once it cyclizes with methyl here versus without methyl here, the RT will read this with 10 times more mutation, right? So not only 10 times of the selectivity, but also 10 times of mutation. So together, she gets 100-fold selectivity. So this is the first method we can really read out the methylated adenosine as a mutation. We take this down to a few nanograms, and now we're using this to do all kinds of interesting studies. Now, I want to take the next 20 minutes and give you sort of a flavor of other modifications. At lunchtime, several people asked me, right? If you look at the messenger RNA, M6A is cool, 0.5%, but don't forget pseudouridine, 0.2 to 0.5%, right? It's the same level of M6A. This one's installed by one enzyme mostly. This one, in the human genome, 13 of them. We know at least 8 are catalytically active, right? And then there are other things. So we decided to look at pseudouridine. How come pseudouridine biology is lacking behind? There's so many papers on M6A because nobody has a good way to sequence pseudouridine. How is pseudouridine sequencing done? Doesn't have very good antibody. Their published antibodies are not very good. There's this CMC method. It's a carbodiimide. This carbodiimide will react with all the nitrogens, right? Will react with all the nitrogens on the nuclear base. And then you treat it under basic conditions, under bicarbonate. Under bicarbonate conditions, the pseudouridine could get deprotonated. So every carbon diimide fall off from nuclear bases except the one on the 3 position of pseudouridine, the one stick here. That's the only one remains on. And what that leads to is a RT stop. The people use the RT stop to read it out. They figured out about 100 sites. There was a Nature paper and Cell paper, and they all work on yeast messenger RNA. They each reported about 100 sites. As we know, the percentage will predict there's at least thousands of sites, right? But they discovered about 100 sites. The same yeast messenger RNA, when you overlay these 2 papers, 5% overlap between the sites they identified, right? So there's lots of false positives, not comprehensive. That was the state of the art in terms of identifying pseudouridine. So lots of people interested in pseudouridine, but they don't know how to find it. I stayed away from this for as long as I could until one day I saw a paper coming out of Bradley Kane, the City Bureau's lab. They found by accident, they acknowledged this, when they were doing bisulfite sequencing, city conditions convert C to U, right, for bisulfite for RNA. they realized the pseudouridine is getting modified. And what we realized, when pseudouridine is getting modified, this leads to a deletion, about 30% of deletion, right? So they identified about 200 sites. And when you do sequencing, this thing will get skipped by the RT enzyme. And Cindy figured out the detailed mechanism is these two complexes, these two adduct that will skip, the RT will skip them. Right? The problem is, this is very hard to repeat because in our hands, to 5 to 30%, it varies all the time, and the maximum yield you get is 30%, right? What you need is 100%. So everybody's stuck, right? Then we realized, gee, this chemistry does not need acid. All the bisulfite sequencing is done at acidic conditions because you have to protonate cytosine to promote that chemistry. We said, why don't we do it in neutral conditions, mild conditions? We get 100% conversion, right? People are so fixed to the protocol, right? All you need is tune your pH to 7, you get 100% conversion, you get 100% deletion. You see there, pseudouridine? If you just sequence it, it's there, but if you perform a bisulfite in neutral conditions, you get 100% deletion. So for the first time, we have a way to detect pseudouridine as a deletion, right? So you can make a library with and without bisulfite, and then when you overlay on each other, look at the deletion. And we went through all the possible sequence contacts, and then we basically can use deletion ratio to calculate the modification fraction for every context. When we apply this to ribosome RNA, we pulled out all the non-ribosome RNA pseudouridine sites, and we pulled out the new sites. And importantly, we can overlay our sequencing fraction versus mass spec detected ones, because mass spectrometry, people have done this. They can isolate every piece of ribosome RNA, use mass spec to look at the modification fraction, and they are pretty much consistent with our results. Now with this, obviously, we went on to ask what's going on in human, how many pseudouridines out there. In cell lines, these are the pseudouridines in different cell lines. I'm not going to bore you, but the point is, we see about 575 sites that are highly modified, over 20%. I come back to this. Lots of different motifs indicating that the different pseudouridine enzymes install this modification. And we can knock down each of them. This is the best sort of the figure I love the most. So this is the without bisulfite treatment. With bisulfite treatment, you can see there's a pseudouridine is about 80% modified. And then we knock out each pseudouridine synthase, and these guys are not installed that pseudouridine because you see the same deletion, but when you knock out 2B1, you restore that signal. So 2B1 instals that pseudouridine, right? So with this experiment, we can assign all the sites to different writer proteins. So this is human HeLa messenger RNA, and we basically can tell you which site is installed by which enzyme. The surprise comes from tissue. I told you when we did the cell lines, we were a little bit disappointed. There's only 570 sites that are 20% or more modified. But when we turned to mouse tissue, we were shocked. Cerebellum, 6,000 sites, which correlates with 0.2 to 0.5%. We would expect several thousand sites, right? And you look at the T cells, thousands of sites. And somehow in cell lines, the pseudouridination is suppressed compared to these tissues, right? Tissues accumulate a lot more pseudouridine sites and at a higher modification fraction. These are cell lines. These are tissues. You can see 40% above, lots of sites. Because of mRNA vaccine, everybody knows pseudouridine. They stabilize RNA. Now we know why. I'm not going to show you the promote translation. We also have clues. There's one interesting biochemistry becomes sort of a mystery for 10 years since its publication. Back in 2011, 2013, research groups at Rochester made an interesting biochemical discovery, never validated inside of the cell. What they discovered was that when you put a pseudouridine to the stop codon, you actually generate a read-through. It's not a stop codon anymore. It recodes. It will recruit the tRNAs to read through the stop codon. right? There's a lot of controversy. Does this really work or not? Because there's a lot of commercial interest. Because if you have a premature stop codons which comes from mutation, this is the way to correct it, right? We went ahead and then we found 106 pseudouridines in native context occur on stop codon. 106 stop codons contain pseudouridine in tissues or cell lines. Not every one of those leads to read-through. Some of these leads to read-through. There's one clear read-through. The percentage is very low. Here's another one. It only occurs to kidney. It doesn't occur to other systems. Well, the pseudouridine occurs to the other system, but only in kidney it leads to read-through. And here's this one, CD52. The read-through ratio is very high. So stop codon pseudouridination does lead to read-through, but it's cell line context dependent, right? There's some sort of a cellular mechanisms to control whether it gets read-through or not, right? There's this additional layer of regulation of translation through stop codon read-through. We confirmed from in vivo evidence. All right, I'm going to finish last part about the 5-methyl-C. So I told you I'm going from biochemistry to chemistry. So DNA has a methylation. 5-methyl-C bisulfite sequencing works very well. Bisulfite sequencing has some problems. It takes 3 hours to run, and it generates lots of DNA damage. But when you apply this to RNAs, it's being a nightmare because bisulfite sequencing works at acidic conditions, 85 degree. 90% of the DNA is getting sort of degraded when you heat it up to 85 degree, cook for 3 hours under pH 4 to 5. RNA, forget about it, it's all gone. So people do this at 65 degree. Later on, they do this at 75 degree, but they have to use a ton of RNA, right? 65 degree, lots of false positives. Right? Has not been really a solution has not emerged. So here's our experiments. These are 2 M5C sites on ribosome RNA. All the red dots are false positive, right? So people have spent several years to solve this problem. They raise temperatures to 75 degrees, treated 3 times. They can start to suppress false positives, but again, they require a ton of RNA, right? Because you have to account for the decay. If you look at the chemistry of this, again, I love this. All this comes from pandemics. Pseudouridine 5C, because we can stay in office, we don't have people bother us. We start to read the literatures and ask, we run this every day, right, bisulfite. Then we can read the literature why, right? Read some old literature. It turned out the bisulfite reaction is actually reversible, right? Makes sense, right? Then you protonated the methyl groups, right? It adds. But then the rate-limiting step is this, the deamination. Now this one, once generated, stays there for 3 hours. This thing stays there, and that leads to depurination and degradation. Right? So this is the problem. Because this step is too slow, and once you generate this, right, it's not conjugated. After you stop bisulfite, you do desulfurination, goes back to stable. Right? So we wondered if we can hit this with a very fast reaction, we probably can avoid all the damage. Because the damage was the product sitting there for 3 hours at 80 degree, 85 degree. So then my senior scientist had an idea. He made it the world's most concentrated bisulfite reagent. Right? Very concentrated. And I hit it at 98 degree instead of 80 degree. Just put a hammer on it. When he put a hammer on it, the reaction is done in 3 minutes. Reaction is done in 3 minutes, not 3 hours. And the methylated C does not change. Right? So better not touch methylated C. Now, with that, you can realize we don't generate much DNA damage anymore. This is with DNA. Now, this is Zymos, like a dog meal, right? There's like all the stuff. But then if you just hit it with few millions, concentrated, high temperature, you don't generate that much DNA damage because reaction is driving all the way to the product, then you quench it. Now, the beauty of this is this is not a fair comparison. This is a single-cell experiment. You don't fragment your DNA. If you do bisulfite sequencing, you realize lots of these sites, because of the CG-rich regenes, you can't denature. They stick. Your false positive is like 30%. Even here, you get a 0.4%. But if you use our method, your false positive is 0.1%. You can clearly see, because we hit it 98 degree, every structure has been denatured, right? Everything becomes single-stranded, so we can run this reaction very fast, and it completely goes to the completion, right, compared to this. So that's DNA. DNA is fine, right? Now we can do this in 10 minutes, right? Your typical bisulfite procedure, 3 hours. In my lab, or we're going to publish this. Now it's 5 to 10 minutes. You can get it done, right? Now our key is to solve RNA problem. Again, we showed that for RNA is the same thing. In 3 minutes, reaction completes, okay? And we don't touch methyl group, right? The methyl ones were not, the methyl still blocks this reaction conditions. And here's the tRNA. Historically, when you do tRNA sequencing, it's highly structured. 65 to 70 degree would not denatured, so you don't get much coverage of tRNA. But now we hit it at 98 degree, and we can clearly see these tRNA sites. And we can knock down the writer proteins and the sign. Here's 3 sites. 2 of those are enzyme 2 sites. When we knock down enzyme 2, see the modification drops, right? And we can assign all these tRNA sites to different writer proteins. And for the first time, we believe we have the true messenger RNA M5C sites. And this is the HeLa cells and HEX cells. We assign the writers because now we can knock them down and perform sequencing again. And for the first time, we can really look at these sites and ask the biology of this. We already know that 5' UTR seems to relate it to translation, and there's a lot more biology we need to learn now with the map at the base resolution with the exact modification fraction. So with that, I'm going to just stop. I hope I showed you some of the biology, and I also showed you that sometimes bringing in some college level chemistry can really help biology, help genomics, and we're matching on to these other modifications just for fun, but I think we probably covered the major ones already, and you know, in the next 5 or 10 years, it will be interesting to monitor the biology following the method that now we can really see where they are. So I just want to acknowledge student postdocs who contributed to the presentation I gave you today. Jun and Xiaoyang did the first 2020 chromatin methylation work, and Jiang Bo and a few others did the demethylase work. And Gui Fang and my collaborator, Professor Song, were the ones we did the plant biology, and lots of the sequencing part were done by Qingdai with Kinka, Arena, and others. So it's sort of a side project in the lab, but they start to build and become quite interesting. And these are my funding agencies and conflict of interest. Thank you very much. I'd be happy to answer any question.
Thanks very much, Chuan. So before everyone starts, I have a question. So I think that all those sequencing methods, chemistry is beautiful to profile all these RNA modifications. But what's your opinion about the nanopore? Do you think eventually we could just use nanopore to look at all the modifications on the same RNA all at once?
Yeah, that's a good question. Nanopore is not very good for M6A so far, right? Nanopore is not accurate enough, but eventually nanopore would be able to do this. But keep in mind that there's so many modifications. Whatever we do here will be easily adaptable to nanopore, right? Because what we're trying to do is to manipulate the modification. If you can manipulate the modification, it's a matter of you can read it off with Illumina sequencing, nanopore, or PacBio.
Cool, thanks. So if you have any questions, please raise your hands. I will go there and try to deliver the microphone because we require everyone to use a microphone to ask questions.
The microphone's on the side.
Yeah. You can come on either side, and I also can come to you to give you the microphone.
Yeah, Laura.
Okay.
Yeah, yeah, I can repeat. Don't worry.
That was very cool. And thanks for changing your talk for me.
Customer designed for you.
Yeah, that was amazing. I actually, but now I have a question about the first part.
Right.
I'm just curious about pluripotency and whether you've looked Because for so early on in embryonic development, right, the cells remain pluripotent and don't necessarily start down the differentiation pathway right away. And I wondered if what you'd found for differentiation is also true and relevant for pluripotency.
Yeah, exactly. So there's 2 layers of that. The first layer I never shared with you even 2 weeks ago. That was actually related to Xiao's work. There are 2 layers of, there's a mRNA level regulation and a chromatin level regulation. mRNA level regulation, we figured that out already.
Right.
So what's going on is most of the pluripotent factor transcripts can be heavily methylated. So when cells decide to differentiate, right, they have to get rid of pluripotent factors. So they use methylation, the YTGDF2 pathway, to decay these transcripts. That allows cells to move to the next cell stage. So it turns out the pluripotent factors are heavily methylated, and this pathway is critical to initiate that differentiation. So in return, we have a way where we're going into translational phase. If you shut down this decay pathway, you can amplify the stem cells, so they won't differentiate. So we can control stem cell self-renewal versus differentiation. Right? So that's the first part. The second part, I also believe the chromatin regulation plays a critical role as well, and we haven't done that much work yet. However, again, the presence of methyl group would suppress the transcription of pluripotent factors, right? And then the demethylase would activate the downstream transcripts, right?
Right. I guess I was asking, too, because of the connection with cancer and the relationship, again, between kind of stem cell pluripotency and cancer cells. So it seems like there could be a lot of connection that your studies will reveal.
Right. Cancer really depends on which cancer hijacked what pathway. It's getting very complicated, but yes. Yeah.
Thank you, Chuan. As always, it's super exciting and very exciting to see the whole story from very beginning to like all these discoveries in the past years. So I have 2 questions. The first one is a simple one. You talked a lot about the line 1 RNA and the modification on there. So how much do you see the modification in alu elements? They are shorter, but they have many more copies in the human genome. They have around like 1 million copies.
Right, there are lots of alu elements. We hide them under the rock. They're hard to map, and I just come back to work on alu elements. So I can tell you this. The first small molecule for METTL3 inhibitors in clinical phase 1 trial, right, as I told you, or maybe not you, others, the trial, is a public information, so I can say this. The trial is not for leukemia, whatever. The trial is actually for immuno-oncology, for immunotherapy. So it turns out M6A play a critical role for double-stranded RNA formation. First thing come to mind are the alu elements. Right? Maybe the methylation occurs to alu elements somehow as a way to tune them. Right? And if you lose the methylation, you probably get more double-stranded RNA. Right? And we also see lots of alu elements, but Irv and others, we do see alu elements, but we haven't really systematically looked at them yet. Yeah. It's also unclear to us it's an intramolecular double-stranded RNA or intermolecular double-stranded RNA. We previously never really thought about this. Whatever data in our sequencing, we probably just filter them. Right.
So the second question is more general. You talked about like engineering the plant, the rice with FTO demethylase. So for a long time, people study health or study animal systems are borrowing the machineries from the plant biology. So how much you would see plant biologists or agricultural scientists are learning from the animal system and engineering plants for agriculture and for sustainable research?
Yeah, I think there was a debate when our paper first came out, right? It's the most downloaded every plant biology reader. There was a debate, and then people realized we're probably the first one to put the animal system into plant. So I don't know if others are doing this. We're doing this at the moment. I haven't, plant biology is severely underfunded, but then it's a very important part, right? I think what we showed is that in plant, this pathway is built in. We're not adding a methyltransferase. We're adding a demethylase. So there's a methylation on chromatin RNA regulates plant growth. We just reverse that, right? And I believe there's a lot more similar pathways, but not exactly identical, right? For instance, we're looking at the ecosystems right now. I personally think that the ecosystems is another really interesting system. The microRNA, siRNA system, right, for instance. Right.
That was terrific, Chuan. I was going to ask you a chemistry question. Because the mechanism of the demethylation involves this aminal and therefore CH abstraction, you might expect a strong kinetic isotope effect if there was CD3 instead of CH3 in that methyl group. And since most SAM biochemistry involves SN2 reactions, any kind of isotope effect would be minimal. It would be a secondary isotope effect. So if you were to, I don't know if you've done this experiment or just would tell me what you think about the thought experiment, replace all of the SAM with CD3 SAM in a cell, would you phenocopy a knockdown in the demethylase?
I think the interesting part would be you have a histone demethylase and other.
Right, there are other demethylases that use this mechanism too. I just wonder how dominant yours
yeah. That's a very interesting question. I think FTO and the TAD systems are the ones that give the most interesting, most major phenotypes. The histone demethylase is also very interesting. So yeah, the concern will be it's very heterogeneous.
Right, but and you of course could use a bump hole strategy to make it more specific.
Right, yeah. Yeah, no, we haven't done that. That's an interesting thought. It does raise an interesting question because people do use these isotope labelings to study kinetics, use mass spec, right? So it may not be the right way to study it because if you put a deuterium, right, the enzymes do not work effectively on them. Now, lots of people use this. They metabolically label, and then they follow their fate through generations by mass spectrometry. But then the question is, yes, CH3 to CD3 change is not like
the same, right?
Right. Yeah. I actually
Certainly
never
not from the
thought
kinetic
about it.
point
I
of view,
reviewed enough of these papers. Never thought about that part. Okay. Right. Yeah.
Yeah, thanks for a fantastic talk, and it was really remarkable to see how big an effect inserting FTO into plants has. And I also liked your model of it being total mRNA abundance that regulates it, and which I suppose is a balance between transcription and RNA decay. So do you think you could further enhance plant growth by inhibiting mRNA decay or mechanisms like that?
Right, that's a good thought. The mechanism of this is not clear, right? It probably opened up the chromatin, become more plastic, right? And then during cell cycle, and then lineage commitment, my hypothesis is that once you're more open, stem cell just stay as a stem cell longer time. That's just my hypothesis. That's why you're accumulating stem cell, right? Exactly what's going on, we don't know. Yeah, that's a possibility. We have lots of ways to perturb this. That's why we started the plant biology at Chicago, because those are the questions we wish to ask. We actually don't know why this leads to accelerated plant root growth. We're not sure it's RNA or it's a chromatin, right? Because you can think about stem cell, right? All the stem cell has a plastic chromatin start to condense when they differentiate, right? Is this to keep stem cell for a longer time, generates more sort of, right? We don't know.
Hi. Thank you very much for your inspiring talk. I have a chemistry question about the last part of your talk. So when you put a hammer on it, to me, the rate limiting step, is after the addition step, right? So why increase the concentration of bisulfite will increase the kinetics of that speed? Or
it's just shifted towards the right?
Yeah, but
It's a bisulfite concentration dependent, right?
But the rate limiting step you mentioned is the deamination step, right?
Sure, yeah.
Oh, okay. Cool, thanks.
You have more sort of a higher concentration on that, right? And now you drive it down, right?
Hi, thanks for the great talk. So I was wondering, the methylation occurs with molecule SAM, and when it gets demethylated, it generates formaldehyde. What's the fate of that formaldehyde molecule, and would this constant process change the pool of C1 molecules, and does that have any physiological effects?
That's a question in the field for a long time, particularly for histone demethylase, right? You have a histone, lots of methyl group. When there's a demethylation generates formaldehyde, what does formaldehyde do, right? We don't know. Formaldehyde will react with cysteines, lysines, right? Generate post-transcriptional modifications. How would that affect chromatin? Yeah, people work on this. Right? Yeah, good question. Just not something we work on, right? Yeah. All right, thank you very much.
Thanks, everyone. Thanks, Chuan.